Hi everyone, this is Mark. This is my first attempt at doing tele-teaching. I'm going to talk a little bit about basic molecular biology, and this is at the request of my wonderful artist friend Ellen in New Jersey, who asked for something more visual. So I'll do two videos, one on basic molecular biology and one on the relevant aspects of the virology. Okay, so to first, basically, or pretty much all organisms, bacteria and bigger, maybe there's some exceptions, I imagine there might be, their genomes are chemically made up of double-stranded DNA molecules, and you've all seen many pictures of DNA molecules, and some organisms, especially bacteria, frequently have circular chromosomes, and many other organisms, including animals and plants and things, have linear chromosomes. Some small organisms may have one chromosome. We have 23 pairs of chromosomes. And the genome contains all the information to make the organism, but it's not like an image. It's not like a blueprint design. It's more like a set of IKEA instructions that have to be built. So there has to be a complicated machinery that knows how to read the genetic code and build the components. And in the genome, there are sections of DNA that actually physically encode carry the information to make proteins. So this is a protein. And proteins do they're the workhorses of the cell. They're the enzymes and structural components of our body, things like collagen, and they're receptors on the surface of the cell, and they're molecules that regulate the function of other molecules or regulate the expression of other genes. And so along the chromosome, we have many, many genes, A, B, C, D, E, and they all have complicated names like ABCA1 and LDLR and SLC25A38, and those are just codes that we use that give some kind of information about what the function of that gene is. And there are some genes that make RNA as their functional component, so that's a fine point. Important, but we don't need to worry about that today. But the key thing is that there's no system, there's no mechanism for reading the DNA directly and turning it into protein. That's not how life evolved on this planet for whatever reason. So there's an intermediate step. So in between the DNA and the protein, there's another set of molecules, which are the RNA molecules, and they function to carry information. So they act as messengers to carry information from the genome through several sets of machinery that eventually will synthesize proteins, which are polymers of amino acids. So glycine, leucine, methionine, tryptophan, histidine, whatever. So every protein is defined by the amino acid sequence, and that amino acid sequence is in turn encoded back in the genome. But the genome is like your hard drive. It has all the software on it. It has code for all of the genes, but any given cell or tissue doesn't need all of the genes to be expressed. It doesn't need all of the possible proteins that the genome can make all the time. So heart cells don't need proteins that are only used in nerve cells, and nerve cells don't need proteins that are only functioning like myosin in muscle cells. So there's a system for deciding in a given cell under a certain set of environmental conditions which genes to turn on and off, and that's a complicated regulatory system. And so if the genome is like a hard drive of your computer that has all the programs on it, if a cell decides that it needs to turn on a certain gene, say gene E, so gene E exists on a particular chromosome in us or somewhere along the bacterial chromosome, and there are signals in the DNA that tell the machinery where gene E starts and stops, and so the machinery has decided it wants to turn on E, and so it makes a copy of E is RNA, and so the RNA in this case is functioning like a copy of the program in dynamic memory. So if you decide you want to run Microsoft Word or Adobe Photoshop or any other software, you double click on the icon and a copy of the program, maybe not all of it, but parts of it, are copied from the hard drive, which is long-term memory, to a dynamic memory, which will eventually be disposed of. And that logic allows the cell to change its state as it needs to, as it develops in an organism, or when the environment changes. If you get a fever, you get an infection, or you've eaten dinner, or whatever, you've exercised a lot, or you've learned something new, you've read a book. Anyway, if different genes need to come on and off, there needs to be a mechanism for turning a gene off again. And you can't destroy genes in the genome because then you could never turn it back on again. So by making an unstable intermediate, which is RNA, you can make the RNA, and then there's a system for degrading the RNA. And so that's how the system can be dynamic. Okay, so that's at a high level how it works. In a little more detail, what happens is here's gene E, and the machinery wants to turn it on and off, and so what it does is it makes a copy of a part of gene E, the part that actually encodes the protein, plus a little bit more. It makes a copy of that in a single-stranded RNA molecule. Okay, so the difference between DNA and RNA is they're chemically very, very similar. The DNA, well, nucleic acids contain bases, which are in DNA are the GATC, in some long complicated sequence, GGTTAAC, and RNA is basically the same except for evolutionary, well, for various reasons. Instead of T, there's a similar base U. So an RNA molecule would be GAUU, CCUU, GAUC, et cetera. So wherever you see a U in RNA, it was coded by a T in the DNA. Okay. So the difference between DNA and RNA is not in the bases. It's that the DNA is actually, it's chemically, it's got a phosphate group and then a sugar, and then a phosphate group, and then a sugar. And the bases are sticking off of the sugars. And the difference is between DNA and RNA is the sugar. In both of them, the sugar is a 5-carbon sugar, so it's a ribose-like sugar. In RNA, it's actually ribose. And to give you a comparison, glucose, which is the sugar that gives us energy, glucose is a 6-carbon sugar. So ribose is a 5-carbon sugar, and in RNA, the sugar is ribose, and in DNA, it's deoxyribose. So there's a particular oxygen in ribose that in DNA is replaced with just a hydrogen. So that's that difference, and that makes a big difference chemically in terms of the chemical function of DNA and RNA, but has no effect on their information content. So that's DNA versus RNA. And this RNA molecule, there are many different kinds of RNA molecules that have different functions, but in this case, an RNA molecule that's carrying information to encode, to drive the production of a particular protein is called a messenger RNA or mRNA. So in us, that's all happening inside the nucleus. And the messenger RNA is exported into the cytoplasm in a regulated process. And there's some chemical rearrangements made on the messenger RNA to mature it, to stabilize it. There's some cutting and pasting that goes on that's not important for today. In bacteria, since they don't have a nucleus encoded by a nucleus included inside of a membrane layer, the messenger RNA is just in the cell, in the bacterial cell. So in the cytoplasm, you've got messenger RNA molecules. And in the cytoplasm, you've got ribosomal subunits, which are floating around looking for messenger RNA molecules. And when they see a messenger RNA molecule, they're just bouncing around, and eventually they bounce into a messenger RNA molecule, and they assemble on it. And then they start looking for a start sequence to start the protein. So the start signals for the protein encoded in an RNA. So inside the RNA, there's also the sequence that will encode the protein, which is a little bit smaller. Those start and stop signals are different than the start and stop signals that the machinery in the nucleus uses to turn the DNA, to make a copy of DNA into RNA. There's another set of start signals. And in particular, the key start signal for starting a protein is the sequence AUG. And so the ribosome is looking around and it sees CCG, AUG, CC, AGC, U, etc. And it's looking along and it finds this AUG and it says, aha, I'm going to start there. As with everything, it's a little more complicated than that. So it sits there, and that does two things. That defines the start signal, but it also defines the reading frame. And if you think about it, the problem is that there's only 4 bases. So if the ribosomes read A, G, U, or C and decided to make that glycine, histidine, tryptophan, serine, you could only code for 4 amino acids. So a 1-letter code doesn't have enough information to code for the 20 amino acids that we have in our proteins. And a 2-letter code isn't good enough either, because a 2-letter code would have 16 possibilities, and that's not enough. But a 3-letter code has 64 possible letters, AAA, AAU, AAC, AAG, AGA, et cetera. There's 64 combinations, and they all are used to provide information. But because there's only 20 amino acids, AA, that's what I use for amino acids, the code is redundant. Some amino acids are coded by multiple different words, which we call codons. And since the code is in threes, by finding the AUG, that also sets the frame, so the ribosome sees the AUG, and then it knows, okay, this is my frame, and so I'm going to read 3 at a time, starting from there, because if it started at the U, it would read UGC, CAG, and that would be different letters, words that would make different amino acids. So the ribosome sees AUG and says, okay, I'm going to read 3, and then the next 3, and then the next 3. And so that defines both the start and what we call the frame. Okay. So here we have the messenger RNA, and the ribosome is sitting on it, and it's sitting over an AUG, and floating around in the cytoplasm also are all the 20 amino acids, but the ribosome doesn't see them directly as amino acids. The amino acids are chemically attached to teeny little RNA molecules called transfer RNAs, or tRNAs. And those are constantly going in and out of the ribosome, and they have different sequences on them. And so the ribosome is waiting until a tRNA goes in that is appropriate for the AUG on the messenger RNA. And when it sees that tRNA, it holds it. And then it reads the next triplet, and in this case, the next triplet was CCA. And so it waits for the right tRNA to come in that would bind to CCA, and it has a different amino acid on it. And the first one is always a methionine. So all our proteins start with a methionine. Sometimes it's removed. So when the next amino acid goes in, the ribosome makes a chemical bond between them, and then it ratchets over one step and releases, and then it can do it again. And so along the messenger RNA, if this is the AUG start codon, you might have a ribosome sitting there that's put one amino acid, and further along the messenger RNA, you've got a ribosome that's made longer, that's made more and more of the protein, and so this is the protein here. And eventually there's a stop signal, and the ribosome knows to stop and release the protein, and then the protein folds up. It may be folding up while it's actually being made, and then the protein goes on to do whatever it needs to do. So that's the basic process, and that's why we came up with, molecular biologists came up with the basic paradigm of DNA to RNA to protein. And for the most part, that holds true with a couple important, well, in us, that basically holds true. But there's a couple exceptions. It turns out that there are some enzymes that are able to make a DNA copy from RNA back to the DNA. And so the DNA to RNA step is actually reversible. But there's no known chemical or biochemical machinery that's ever been found in any other organism that can take the protein sequence and code it back into RNA. It's not unimaginable. The amino acids in the protein are there. If you looked at amino acid, it would be methionine, and then, glycine, histidine, serine. And you could imagine a machinery which says, well, methionine, that's an AUG, so I'll make an RNA with AUG. And then glycine, I've got several different words that could make glycine, but I'll pick one of them, GGG. Maybe that's it, I'm not sure. And so you could make an RNA molecule in reverse, which would reverse code the protein. And that would actually, in principle, could allow for Lamarckian-style adaptive transmitted inherited evolution. But there's no mechanism known for that, so it's never been observed. So there's no reversing of this step. Okay, that's it for Basic Molecular Biology 101. Thanks for listening. In the next one, I'll apply this to viruses.